All right, let's jump right in. Today, our explainer is going to completely demystify the wild, fascinating world of the RNA-seq pipeline. We're going to trace the chronological journey of a single biological molecule, literally a microscopic speck, and follow it from the wet lab all the way to a data scientist's screen where it becomes millions of lines of code. If you've ever found yourself wondering how invisible biological matter actually transforms into actionable, statistical data, well, you're in exactly the right place. So here's our roadmap for the day. We've got a very clear step-by-step path from raw biology to a neat mathematical count matrix. First up, we'll decode gene expression. Second, the physical sequencing workflow. Third, reading the FASTQ file. Fourth, mapping the genomic puzzle. And finally, the final countdown to our data matrix. Let's do this.
Starting with Step 1, decoding gene expression, and the core biological problem we're trying to solve.
Now, when we study gene expression, what we're really doing is measuring messenger RNA. We want to see exactly which genes are actively being turned on inside a cell. But as you probably know, biology is really as simple as one gene equals one protein, and that's entirely due to something called alternative splicing. It's kind of like editing a movie. You shoot the exact same scenes, which represent our exons, but you can edit them together in different ways to get two completely different endings. During alternative splicing, specific exons of a gene might get included or completely left out of the mRNA. So a single gene can actually code for multiple distinct proteins that do totally different things in the body. And that adds an incredible layer of complexity to the mRNA we're trying to measure. Okay, moving on to Step 2, the sequencing workflow. Let's take a look at the physical solution. Let's walk through the wet lab library preparation pipeline. It's basically a microscopic assembly line. We start by collecting our biological samples and capturing that super delicate mRNA. Then we have to fragment it. Current sequencers just can't swallow whole transcripts, so we chemically or mechanically chop them down into bite-sized chunks, usually around 300 to 500 base pairs. After that, we do reverse transcription. Since the dominant sequencers out there only read double-stranded DNA, we have to convert our single-stranded mRNA into robust, machine-readable complementary DNA, or cDNA. Next up, we ligate adapters onto the ends. Think of these as platform-specific barcodes or handles that the sequencing machine will recognize. Then, we use PCR amplification to beef up the concentration. And finally, we load this beautifully amplified library onto the machine and we sequence it. Now, here's where it gets really interesting. We have two main ways to actually sequence those fragments. There's single-end sequencing, which just reads one end, either the 3' or the 5' end of the cDNA, and then we have paired-end sequencing, where the machine reads both ends. Paired-end sequencing is fantastic because it effectively gives us double the data per fragment. And as you'll see, having double the data is absolutely crucial for improving how well we can align those reads later on in the pipeline. All right, so we've built the library, the machine has run its sequencing by synthesis magic, and the physical run is over. Which begs the question, what does this sequence data actually look like? Because this is the exact moment we pivot away from physical biology and robotic machinery straight into the digital realm of data science. That brings us to Section 3, reading the FASTQ file, or what I like to call the digital funnel. Our physical molecules have officially been transformed into massive gigabase-sized digital text files, packed with millions upon millions of what we call reads. The standard file format you're going to deal with here is the FASTQ file. Let's break down the anatomy of just a single read within that file. Every read takes up exactly 4 lines of text, no more, no less. Line 1 is the sequence identifier, and it always kicks off with an @ symbol. Line 2 is the actual meat of it, the biological sequence itself, all your A's, C's, T's, and G's. Line 3 is just a secondary identifier, starting with a plus sign. But line 4? Line 4 is incredibly important. These are the ASCII quality scores. See, as the machine reads the fragment, it's translating light signals into base calls, but it's also recording exactly how confident it is in every single one of those calls, and it encodes that confidence as ASCII characters just to save storage space. So those ASCII characters are essentially a secret code for mathematical Phred scores. To put that into perspective, a Phred score logarithmically relates to the probability that a base call is flat out wrong. A score of 10, that means 90% accuracy. A score of 20 gets you to 99% accuracy. But look at a Phred score of 30. That equates to a reassuring 99.9% accuracy rate. 30 is the magic number. It's the gold standard threshold we look for to know for sure that we can actually trust our data. But before we can actually analyze this data, we've got to clean it up. We do this in a pre-processing phase using quality control tools like FastQC. We trim away any of those leftover adapter sequences, we snip off deteriorating bases at the ends of reads, and we filter out the low-quality stuff. But hold on, here is a massive point to remember. In RNA-seq, we never ever remove duplicate reads. Why? Because unlike DNA sequencing, where a duplicate almost certainly means a PCR error, in RNA-seq, duplicates are totally natural for highly expressed genes. If a specific gene is just churning out thousands of copies of mRNA, we absolutely expect to see identical reads. Deleting them would literally destroy our biological data.
Section 4, mapping the genomic puzzle and finding the origin of those reads.
So right now, our millions of clean text sequences are just floating around as disconnected fragments. We have to map them back to their genomic features. It is quite literally like trying to assemble a billion-piece jigsaw puzzle, and we need the picture on the box lid as a master guide. We actually have two different guides we can choose from. The first is a reference genome. This is the whole shebang, the full representative genetic sequence of the species. But mapping to a genome requires complex spliced alignment. Remember alternative splicing? A read might span an X-sized intron, meaning its two halves could map thousands of base pairs apart on the genome. It's tricky. Option 2 is mapping to a reference transcriptome. This only contains known, already spliced isoforms. Because transcripts are way shorter than the full genome, this method is incredibly fast and memory efficient. Though it's worth noting, using a transcriptome can sometimes mistakenly flag intronic reads as spurious expression. And it's not just the guide that differs, it's how we use it. Traditional alignment-based workflows are computationally heavy. They try to pin down the exact genomic coordinates where a read maps, explicitly dealing with those messy spliced junctions. On the flip side, we have modern alignment-free workflows. These are what we call lightweight and fast. Instead of agonizing over exact coordinates, they use probabilistic models to simply ask, hey, which transcript is this read compatible with? These modern lightweight approaches pull this off by using something called K-mers. A K-mer is basically just a short sequence of nucleotides of a specific length, K. The software chops up our reference guide into a highly searchable index of these K-mers. So when it looks at a read, it scans for matching K-mers to establish transcript compatibility. This creates what are known as equivalence classes. And honestly, it's a brilliant computational shortcut. It effortlessly handles multi-mapping reads. If a read shares K-mers with 3 different spliced isoforms, the model just groups them into an equivalence class, rather than having a meltdown trying to force it into one exact genomic coordinate. Which brings us to Section 5, the final countdown, abundance quantification. Okay, so we've mapped our puzzle pieces, but now we have to translate those mappings into hard, actionable numbers that tell us how strongly a gene is actually being expressed. We can do this through counting or estimation. Direct counting is exactly what it sounds like, a rigid integer count of fragments that definitively map to a specific gene level. It's the traditional method. But estimation, that's where the whole field is moving. Modern tools use statistical models, like the expectation maximization or EM algorithm, to probabilistically assign fragments to transcripts. It's incredibly elegant because it smoothly resolves all that ambiguity with multi-mapping reads we just talked about. It gives us a highly accurate estimated abundance instead of a rigid and sometimes flawed whole number count. But wait, we're not done. Raw counts, even the estimated ones, can be incredibly misleading. If we just run with the raw numbers, we smack right into 2 massive technical biases. Bias number 1, sequencing depth. If you simply run the machine longer on sample A compared to sample B, sample A is naturally going to have higher counts for everything, even if the actual biological expression is perfectly identical. Bias number 2, transcript length. Think back to library prep when we fragmented our molecules into 300 to 500 base pair chunks. A massive, long transcript is going to break into way more fragments than a short one, which artificially inflates its read count. We absolutely have to mathematically correct for both of these issues. Enter TPM, transcripts per million. This right here is the ultimate equalizer in RNA-seq analysis. The math is brilliant. First, it divides the read counts by the length of each specific transcript, which normalizes for feature length. Then, it divides those length-normalized values by the total sum of all the values in the sample, taking care of sequencing depth. The result? It transforms our raw bias data into a clean, level playing field. TPM lets us fairly and accurately compare gene expression across completely different biological samples and completely different sequencing runs. And boom, here we are. We've officially reached the destination. Once our abundances are quantified and normalized, we pack them into a tidy little count matrix. It's a beautifully simple setup. All of our specific genes span the rows, and all of our different biological samples span the columns. This one matrix is the absolute bedrock foundation for any downstream biological analysis we could ever want to do. Just take a second to think about what we've actually accomplished here today. We took invisible, single-stranded molecules from a cell, chopped them up, converted them to DNL, read them as microscopic flashes of light, mapped them against a billion-letter genome, and corrected the math to build a perfect digital matrix. It's an absolutely wild journey. But the real question is, what groundbreaking biological secrets, what disease cures or massive genetic discoveries are hiding inside those columns and rows? Well, that's exactly what data scientists get to extract next. Thanks so much for joining me on this explainer, and as always, keep questioning the data.